Hello. So in this lecture, we will cover some environmental factors that are controlling bioaerosol concentrations in the indoor environment. Because if we know these things, we will be able to better able to control these things as well. The bioaerosol exposure in the indoor environment. So some of the environmental factors that control bioaerosol concentrations would be the moisture content of the building, the relative humidity, basically, the temperature, then things such as the air exchange rate and also the human activities. So some of the things we have already covered in terms of air exchange rates and the associated calculations, and we have other things as well which we will look a bit more, and we will also look at a case study on this. So relative humidity, the indoor fungus level are almost, it has been found, lower than the outdoor count when we have lower humidity in the indoor area. So observations have supported that having a more humid environment sort of proliferates the growth of the fungus inside. In general, it is said that the indoor fungus development is quite influenced by the moisture and the presence of, let us say, sources that will allow the growth of the fungus. So in that way, once we know that the concentrations and the growth of fungus is influenced by the moisture, we can devise some methods, or we can devise strategies to control fungus for limiting or eliminating the fungal development. And so these would include controlling the moisture levels and lowering the indoor organic pollutant concentration or the inorganic pollutant load. The fungal count can therefore be decreased, for example, by having mechanical ventilation. So as with the chemical pollutants, if we provide more ventilation, we can allow for the flushing of the indoor air, and therefore the fungus will be able to be flushed out and the air inside will be replaced by the cleaner air. So in this way the fungal count can be decreased. Then, as we have seen that moisture or relative humidity inside is positively associated with the fungal concentration indoors, one can have the forced air heating, which will sort of limit the moisture content of the air. Then one can also have dehumidifiers, so that the relative humidity is reduced. One can have air filters, and air filters in the air conditioners also. So that way we can control, so air filters and the same filtration mechanisms in the AC, and we can have reduced fungal counts. In general, the control of excessive moisture and the consequent fungal development will depend upon the design of the building, the construction, and the upkeep of the building envelope. Now the other factor is the temperatures. So all the bacteria or the microbes have an optimum temperature for growth, which means if the typical indoor temperature is somewhere around 30 or plus or minus, those are optimum conditions or near optimum conditions for the growth. However, if the temperature is much high, then the bacteria or the microbe may not survive. The temperature and the survival depends on which microbe we are talking about. So the bioaerosols may be influenced by the temperature of the indoor air. But if we know that if we increase the temperature more, maybe they will not be able to survive. So thermal energy has also been used to control the bioaerosols through moist heat, which is using steam under pressure, and dry heat, which would be the high temperature without the moisture, and maybe thermal energy released by electrical heating coils in a specialized environment, not the heating coil in the indoor air, may be helpful in the controlling bioaerosols. For example, exposure to temperature of 100 to 140 Celsius for even sub-seconds can inactivate the airborne bacteria. It can probably decrease the size of the fungal aerosol, because if these aerosols acted as a condensation nuclei, increasing the temperature may vaporize the things that were condensed, and simultaneously also the concentrations can be decreased. For example, this has been observed for these compounds. The proteins may also get denaturized and the microorganism may get damaged when it is exposed to very high temperature. Here I will present a case study, and this case study will show that the dependence on humidity and temperature was how it was analyzed. So this case study is on the analysis of ambient concentration of airborne microbes and the endotoxins in rural households of South India. The motivation was that almost 69% of population of India was generally lives in the rural places. Also, due to the poor socio-economic status, the rural areas may be overcrowded. In some places, depending on how the houses are, if they are very small houses, the houses may be ill ventilated. And this overcrowdedness and ill ventilation may lead to an accumulation of chemical pollutants in the indoor air, accumulation of vermins and the microbes. So the objectives of this study was, in a rural setting in South India, to characterize the bioaerosols in the indoor air, to observe the endotoxins that are in the particulate matter in this indoor air, and to obtain a better understanding of the bioaerosols in both the indoor and outdoor regions of this households. So the study design involved analysis of bacteria and fungi and analysis of endotoxins. So first thing would be the sampling site selection, then followed by the sampling of indoor air for bioaerosols, sampling of outdoor air for bioaerosol. Once this sampling is completed, the samples would be analyzed by cultural and molecular analysis. An assessment of the pathogenicity would also be made, and the effects of indoor ecology, meaning what is the human occupancy condition inside the houses, what is the pets, what kind of things are being used for cooking, what is the temperature, what is the relative humidity. So this will be measured or noted, and the bacterial and fungal concentration in the indoor environment will be checked for their association with the indoor ecology using regression methods. Similarly, for endotoxins, the particulate matter will be collected. From this collected sample, the particle bound endotoxin will be analyzed using LAL assay and the endotoxin concentration determined. Once these concentrations are determined, again through these regression methods, we will check how these concentrations were influenced by temperature, humidity, even the presence of outdoor or the composition of outdoor air, and the human activity, presence of pets, and such variables. The sampling area was the village settings in Telangana around this region. Around 35 houses were then selected in this village in South India, and this is an illustration of how the houses may look from the outside. So there was like a window, a door, a window, and such things. They do not look to be like huge houses with, for example, huge houses which may have courtyard in between. Mostly they are houses that are smaller in sizes and have not very high level of ventilation. Samples were collected in these houses, mostly from the living rooms, and the window presence, door presence, whether they were open and closed, these kind of parameters were also noted. This illustrates the results. First is the microbial concentrations. On this figure, we have the bacterial concentrations in colony forming units per meter cube. So indoor concentrations, the median value was around 750 colony forming units per meter cube. The 25th and the 75th percentiles were around 300 and 1000, and these were the larger ranges, 95 percentile ranges. The concentrations of these bacteria indoors was significantly higher than outdoors. We can see easily the median was above 1000. Likewise, we have the figure here of fungal concentrations in colony forming units per meter cube. Here it was the opposite. The outdoor concentrations with a median of around 700 CFU per meter cube, the outdoor concentrations were actually higher than the indoor concentrations, median around 500 CFU per meter cube. So this is summarized here that indoor air, the concentrations were 1031 as median, and the range percentile values. Same for the fungi. So for bacteria, indoor air had higher concentrations than outdoor air, but for fungi, indoor air had bit lower concentration than the outdoor air. This is the result on the bacteria that were found. In summary, we see here 3,748 colonies were observed with 79 bacterial species. The Bacilli family was the most abundant in both the indoor and outdoor air. The parameters that were also calculated were the relative abundance, which means the evenness of individual species among other species in a community, and the species richness, which is the number of individuals per species in a community. Human-oriented species such as Microbacteriaceae, Staphylococcaceae, and Enterococci were also observed. Additionally, the species found in fruits and food, Acetobacteriaceae, were also found. If we see at the diversity, the indoor air had again Microbacteriaceae, like we said earlier, then these ones which are actually quite associated with food and fruits, and this one which is a human associated species. So this naturally suggests, and we had Bacilliaceae, this pink color, which was the highly abundant family. So we see here that already we are seeing human oriented species in indoor and outdoor air. Let's see about the fungi. In fungi also, 2,681 colonies were observed with 67 fungal species. Trichomonaceae was found very abundant in indoors as well as outdoors, the dark blue one. And then there were other species also that were found in varying amounts, and this figure can also show how indoor and outdoor air can be different. For example, Hypoxylaceae proportion is much higher indoors compared to outdoors, whereas Sodariaceae's proportion is quite higher outdoors compared to indoors. So same species could be found, and this Xylariaceae was seen outdoors in reasonable proportions but is not seen indoors. So the community composition itself can change quite a bit in indoor compared to outdoor environment, and their relative proportions can also change quite a bit in indoor versus the outdoor environment. This is a figure, or this table basically plots the dependence of the indoor bacterial concentration and the indoor fungal concentration as a function of many parameters such as outdoor bacterial concentration, outdoor fungal concentration, outdoor relative humidity, outdoor temperature, indoor bacterial concentration will not influence, indoor relative humidity, indoor temperature, number of inhabitants, number of rooms, the presence and absence of pets, the age of homes, and the type of cooking fuels. So first we will look at the indoor bacterial concentration. So it was found that indoor bacterial concentration was significantly correlated with the outdoor bacterial concentration. So this makes sense, as we have already studied, the ventilation in houses will lead to the bringing of outdoor air to indoor air. So there will be a correlation between outdoor and indoor air bacterial concentrations to some extent. The indoor bacterial concentration was not related to the outdoor fungal concentration, which is intuitive, because how will bacterial concentration relate to fungal concentration? The indoor bacterial concentration was not related with the outdoor humidity, not related with outdoor temperature, not related with indoor humidity in this particular setting. However, it was significantly correlated with the indoor temperature. This would be because the temperature of the rooms where the study was carried out was around between 25 and 40, mostly 25 and 35 Celsius, which are the optimal sort of conditions for the growth of bacteria. And therefore, there was a correlation between the bacterial concentration and the indoor temperature. While the number of inhabitants in this study was not significantly correlated, and the number of rooms and the presence of absence of pets was also not significantly correlated. So they may have been the sources, but not influencing the bacterial concentration very much. There was a significant correlation with the age of the house, because the older the house, the older it may have bacterial growth. So there was a positive correlation. Likewise, compared to LPG, the biomass was also the burning of biomass fuel, mostly cow dung and wood, was influencing the bacterial concentration, most likely because these were also providing the particulate matter surfaces for bacteria to grow. When it comes to the indoor fungal concentration, in this case it was found that the indoor fungal concentrations were significantly correlated with only the outdoor fungal concentration and nothing else. This suggested that in terms of fungi, the impact of outdoor air fungal concentration was much higher in influencing the indoor fungal concentration, whereas the indoor parameters were not impacting this indoor fungal concentration that much. So already we can see that some of the ecological, indoor ecology, indoor meteorology parameters are influencing, and this influence is different between bacteria and fungi. The next, we also looked at the indoor air endotoxin concentrations, with the median concentrations being 4.6 endotoxin units and minimum and maximum. And these indoor endotoxin concentrations were positively correlated with the indoor bacterial concentration and negatively correlated with the relative humidity indoors, and then positively correlated with the biomass. So endotoxins are basically released from the outer cell wall of bacteria. So if we have more bacteria, we will have more endotoxins. So there was a positive correlation. There was a negative correlation with indoor humidity, because if the humidity decreases, the cell will be sort of killed, and when the bacteria will not survive because it will sort of desiccate. If the bacteria doesn't survive, it might break, it might just get ruptured, and more endotoxins will be released, which are basically the part of the bacterial structure. Then the association with positive biomass indicates that basically more particulate matter has been released and more chance of endotoxin to be attached to this particulate matter. So indoor bacterial concentration and this meteorological factors played a significant role in determining the concentrations of endotoxin. So we see that depending on study to study, but it does appear that the outdoor concentrations, indoor concentrations, and the indoor meteorological and ecological settings influence the indoor microbial, meaning indoor bioaerosol, bacteria, fungi, endotoxin concentrations substantially. So the outcomes of this study were that the bioaerosol concentration and composition was varying among all the sites. The bacterial concentration indoor were more than outdoor. The fungal concentration in outdoor air were more than in the indoor air. The indoor ecology parameters were a significant contributor. For example, for bacteria, temperature, humidity, number of inhabitants, age of homes, types of fuels. Also the number of inhabitants, because the more the number of inhabitants, the more bacteria may be, microbes may be emitted. Outdoor source also acted as a potential source to indoor air microbial load. The endotoxin concentrations varied, and the indoor bacterial concentrations were a significant predictor variable, which makes sense because the more the bacteria, the more the possibility of the endotoxin being released. So once we see that, now we can also list out some other parameters such as building replacement, which is the replacement building ventilation. Building ventilation being the replacement of indoor air with outdoor air. Of course, the ventilation is needed to limit accumulation of the chemical contaminants and the bioeffluents from human occupants, so the bacteria or microbial load emitted by humans. Ventilation also can be used to limit the concentrations of pollutants from the indoor sources. Then we have different types of ventilation which can help remove the biological contaminant, such as local exhaust ventilation and the general ventilation. What does it mean? The local exhaust ventilation would mean the removal of contaminants generated within a space and using the various design of capture device. So fume hoods or localized air blowers will remove the contaminant from a very local space within the room. This exhaust ventilation takes place as close to the surface of the contaminant generation as possible. Some example we already know, for example, kitchen hood, bathroom exhaust of sewage injector pumps, which are localized in their effects. General ventilation is a general ventilation. It can reduce and remove airborne contaminants using either the method of dilution or the method of displacement. The dilution or mixing ventilation will try to mix the contaminated air with clean air, and therefore it will dilute the contaminated air, so to speak, and the concentration of the contaminant will become lower. And we would ideally want to provide in a way that we reach accepted safe level. Conditioned air can be supplied at relatively high velocities from air diffusers. For example, those located at ceiling height, and sometimes the return air can also be at the ceiling height, and we can flush the indoor air with the outdoor air. The displacement ventilation basically tries to push the contaminated air away from the breathing zone in as close to a laminar directed flow as possible. One can also use and help in this kind of ventilation the plumes from occupants, electric equipment and other heat sources. This is an example of like the dilution and mixing ventilation. We may have the air coming in here and sort of it flushes all the indoor air, mixes with indoor air and therefore dilutes it, and then we have the exhaust. This may be an example of displacement ventilation. So air through, let us say, a clean filter is blowing very close to the inhabitant and is sort of displacing the dirty air or contaminated air away from the inhabitant and replacing it with the fresh air, and then these bacteria or microbes can somehow go a bit up and go out from the exhaust. So here the idea would be to dilute this. This is the occupied zone. So dilute the occupied zone, and here the idea is to displace the air away from the occupied zone. Removal of bioaerosols by filtration through one of these filters, MERV 13.7, which achieve different levels of efficiency for different levels of particle sizes, combined with the outdoor air, has been shown to achieve high removal efficiencies of bioaerosols. For example, filtered together with 5% outdoor air, almost 90%. With 20% outdoor air, also almost 90%. With 15%, also 9. So up to 90% of bioaerosols can be removed. When we have a filter with less removal efficiency, such as MERV 7 rating, the control is almost only about 50%. So having filters and outdoor air combination can help and reduce the bioaerosol concentration indoors. The next is the occupancy, and referring to a case study on a class in a school environment, it was found that the number of total indoor bacteria were 1700 CFU per meter cube in a classroom, even when there was no occupant at the time of analysis. The concentrations were much higher in the washroom, and 13 inhabitants were there. Likewise, they were higher in the canteen area, and 35 inhabitants were there. Library, the concentrations were lower, and then in the lab there were concentrations. In the seminar, the concentrations were also about 1695, quite close to the concentration in the classroom, even when there was no inhabitant in the seminar hall. So despite the fact the classroom and the seminar hall was empty at the time of analysis, there was indoor bacterial count. But this could be because in general those places are inhabited or frequented by students and teachers. So they would have emitted some bioaerosols. Also, some bioaerosols may have come from outside to inside, and so we would have bacterial count in the indoor air of those rooms. Also, it could be the bacteria would be because the cleaning was done before the lab or the class, and then the students came, and then they sort of replenished or they contributed to the bacterial load, as I just said. Also, here it was seen that the halls were kept closed, and in that case the circulation was limited, so whatever was emitted inside remained inside, and therefore the bacterial count was there. So it was also found that the average bioaerosol count in seminar hall was somehow associated with the enclosed indoor space inadequate cleaning schedule, meaning mostly that the PM matter would not have been cleaned, which would allow more bacteria or endotoxins to associate with the PM matter. The enclosed indoor spaces and lack of circulation of air. So this allowed for the buildup of microbes. So in summary, in this lecture we have obtained information on the bioaerosols in the indoor environment, the sources of bioaerosols to indoor air, the health effects, and some factors that control bioaerosol concentration in indoor air, such as humidity, temperature, ventilation, and occupancy.